Okay, I'm Sandra Arias. I've been working with the Nano Lab, Nanotech in Thailand, and what I'm doing is the synthesis of molybdenum dioxide to catalyze the production of octanoic acid. What I do is I synthesize catalysts using simple terms such as hydrothermal. It's a one-pot synthesis, which makes it very easy to control. And there are other ways you can synthesize molybdenum dioxide, which is through impact, but that's something else. So there are three ways that octanoic could have been. So through the oxygenation process, it could have gone through hydrogenation, decarboxylation or decarbonylation. It's not on there, but it's supposed to. So the reason why I chose molybdenum dioxide, it's because it has very similar structure to molybdenum sulfide, which is the traditional catalyst. We have been using molybdenum sulfide in our labs for a very long time, and it has no chance of sulfur contamination. The reaction is to produce molybdenum dioxide is very simple. In the one-pot synthesis, we put in ammonium molybdate tetrahydrate with vitamin C, and this is a basic setup. So the product that we got, it's in powder form. I extract it through centrifuge. So going through XRD, I know that comparing the result with other database, it is indeed molybdenum dioxide, but I vary the hours I use to produce the molybdenum dioxide to 4 hours, 7 hours, 3 hours, and even 1 hour. And it seems like the lower the time we use, the less surface area we have. So by increasing the time, we can see that the greater the surface area, that's more efficient as it's a catalyst. So after getting molybdenum dioxide, I put it into reaction with the octanoic acid, and this is what we got. To be a good catalyst, after the reaction, it should be reusable, which means that it should be able to extract out of the octanoic acid product. But in this case, it pretty much diffuses into the octanoic acid, making the product like black. Yeah, so but I put the product into GC, and what I got wasn't what I expected. I put the peak one is hexane, and this one is octanoic acid, and these are all the unknowns that when looking through the database, I couldn't find what it was. And this is octane, what I was looking for. Yeah, so it's an ongoing research, and I'll conduct some more experiments.
Hi, my name is Sandra Arias. This is my poster. I'm presenting a work titled Bioactive Hydrogel, membrane based on bacterial nanocellulose for supporting neurovascular aneurysm neck closure. This project trying to get a better approach for treating the aneurysms, cerebral aneurysms. So we are using a hydrogel that is produced by a bacteria, and these hydrogels is pouring in the neck of the aneurysms, and we are trying to attract muscle cells to this area and start closing that defect. So in this work, we did some mechanical testing of the hydrogel, and we found that these materials is very close to the properties of the bacterial wall. So it's very suitable for this kind of applications. Okay, thank you.
Hi, I'm Kimberly Kidada from West Virginia University. I'm here to talk about our REN at WVU and NanoSafe graduate fellowships. Both of these graduate fellowships are in the area of nanotechnology. Our REN at WVU is an IGER program, and it focuses in nanotoxicology. Our NanoSafe program focuses on nanobiosensing. Both of these programs have graduate students, and both groups are working together synergistically in the area of nanotechnology. So our IGER program specifically focuses in nanotoxicology, as I mentioned. We have about 14 graduate students in our REN at WVU program, and we have about 13 students in our NanoSafe program. And again, the groups work together on different activities, interdisciplinary research, science communications, an innovation challenge, and team building activities. So again, in terms of our interdisciplinary research, each student has 2 advisors. In many cases, those advisors are from vastly different programs, for instance, physics and pharmacy, or chemistry and engineering, chemical engineering and pharmacy. So we have a variety of researchers working together throughout West Virginia University on these projects and helping these students. In terms of science communications, one of our programs that we're working on with our NanoSafe and IGER programs is called Science on Tap. So the students are organizing lectures for the community. Our next upcoming lecture is in art conservation. So an expert in art conservation will present at a local brewery. They're encouraged to have a social atmosphere and to present to the wide variety community. Our last Science on Tap had about 75 people from the public, so it's a well-attended event. We also work very heavily with outreach activities throughout West Virginia, probably about 12 of those per year. In terms of our Innovation Challenge, students at the IGER NanoSafe program have the opportunity to create a business plan and gain about $15,000 seed money for their programs. Also in terms of team building, so that we can create a cohesive network amongst our students, we're regularly doing team building activities. We have some pictures here, but high ropes course, low ropes course, and all kinds of different team building activities.
Hi, my name's Matthew Martin, and this poster that I'm presenting is based on some experiments I did when I was a postdoc at the National Institute of Standards and Technology. Currently, I'm an assistant professor of physics at Khalifa University in the United Arab Emirates, and this work was motivated because there's a lot of interest in the stability of nanoparticles in environmental, biological conditions, but it's very difficult to quantify between the loss of nanoparticles due to dissolution and aggregation. But it's very important to quantify the processes because they compete against each other, and they both lead to a loss in silver, or in this case silver, or material from a metal core of a nanoparticle. So what I did was I took a highly relevant nanoparticle material, silver, and I coated it with a highly protective protein coating. So what I have is I have a rusting metal core protected by a protein coating that buffers the nanoparticle from the solution environment. And then what I do is I stick it in an X-ray beam as it's dissolving in acid. And what you see is if you add just a little bit of acid, you don't change the solution that much. Your measurements don't change over almost 8-9 hours. If you add a little bit more acid, you see that the particles, they'll start to scatter more light, or more X-rays, which means they're starting to aggregate and cluster together in this sort of base structure. Now, if you add a ridiculous amount of acid, where you completely rip open the protein coating, the particles will aggregate, dissolve, and sediment all at the same time. And so it becomes very challenging to separate the loss of material due to aggregation, dissolution, and sedimentation. But if you're careful, you can look at simple quantities like the diameter of a particle. And if a particle doesn't change its diameter, you have aggregate growth, but you have loss of silver over time. It leads you to believe that dissolution doesn't work like peeling back the layers of an onion, but it works like popping bubbles. And that's what actually happens. When you add a lot of acid to this solution, a particle surface that has a globular protein, this protein might unravel, and it would cause a patchy surface coating, and you'd have a vulnerable particle. And if you're very careful with TEM measurements, you can actually see that. You can see that where either there wasn't a protein, there was a vacancy, or where you've unraveled a protein and now you have access to the metal core, the silver will dissolve over time. And if you make the TEM guys a little bit upset, you can add a little bit of water to your sample, and you can see that process happening.
That's why we went with
over time. And so this leads us to this overall model where if you add just a little bit of acid, the particles, due to fluctuations in solution chemistry, they might lose atoms and dissolve. But if you add a ridiculous amount of acid, if you really pull this protein apart, you're going to cluster the particles together, and the material will leak out particle by particle. So instead of, as I said before, instead of peeling back the layers of onions, you're clustering grapes together and then picking one off the vine one by one. And so I want to expand this work to not just proteins, but biocompatible polymers, polymers used in the food industry, and also any other coating that someone can find useful. We chose a very messy thing, protein that likes to aggregate, to prove that we can do this in a very complex system. And so that's the work, and we hope to expand it in the future.
Oh, my name is Santiago. I'm from the University of Florida at the Siemens Lab. And basically my research is on how induced pluripotent stem cells derived vascular smooth muscle cells from hypertensive patients behave after the addition of a contractile drug. So basically, different contractile drugs affect people differently. Reason hence, when you take a cell from the skin or the blood vessel and you differentiate it specifically into vascular smooth muscle cell, you maintain that genetic information from the patient. So my research is on making a very sensitive mechanism that picks up the force that the cell produces when you add a drug. This process consists on making a polyacrylamide gel, and over it makes a very, very thin gel with fluorescent bits. When you seed the gel, after functionalization of the gel, the cell it's gonna pull on the gel. It's gonna pull on the gel along with the bits. So by knowing where the bit is and where the bit was, you're able to quantify the displacement of the bit. Because you know the specifics of the gel, so you know the stiffness, which is the Young's modulus, and the Poisson's ratio, you can quantify the strain energy and the force that the gel had to produce to move that bit. Several hiccups that we have found to be able to make this process a lot more specific is increasing the bit concentration. That way you can pick up small changes in the force, and making the bits a lot smaller, because at least the bits we're using right now are 3.2 micrometers, when an average a cell it's about 10 micrometers. So the smaller the gel, the smaller changes you're going to be able to pick up. We're pushing this experiment to be as close to the physiology of the human body. So we're going to be looking into making the stiffness of the gel around 20 kilopascal, while right now our experiment shows results for a gel that is about 150 kilopascal. And Thank you.
Hi, my name is Nicholas Calvo. I'll be starting my first year as a PhD student at University of Florida in the Gator Bait Laboratory. My poster here is on dynamic cell culture of 3D cardiac microtissues. So basically, there's predicted to be 1 million incidents of myocardial infarction or heart attacks in the next year in the US, and 30% of which are recurrent incidents. And that amounts to about $200 billion a year in direct and indirect costs. So this is kind of an important clinical diagnosis thing. So we're trying to use induced pluripotent stem cells that are patient-specific from people with cardiovascular disease to analyze how stem cell therapy, cell and gene therapy treatments can help wound healing in these areas. So kind of the overarching idea is we take these stem cells that we get from the hospital, we'll differentiate them into cardiomyocytes, induce them into scar tissue, and then take these stem cells and try to see how it interacts with the scar tissue. So to do that, we've started developing these mechanical systems, first of which is a low throughput stretching system that fits perfectly in a cell culture plate for an in vitro microscope. So this allows us the benefit of doing real-time imaging on how stretching these cell constructs, whether it be cardiac tissue or the actual infarct tissue, how it's affected by nanoparticle uptake or these stem cell treatments, what paracrine systems are activated. It just kind of gives you a real-time testing platform in an inverted microscope. These high-throughput stretching systems utilize vacuum to perform their stretching. So you can see here that there's a thin silicone membrane here that your cells are plated on, and when your vacuum's pulled around the posts, you get stretching of the cells biaxially. So using that, they're in a 12 well and 96 well plate fashion, so you can do very high throughput analysis of cells biochemically and functionally using this system here. So we're also trying to implement these 3D constructs for stretching. Everything in the body is three-dimensional. So by embedding cells in these 3D collagen constructs and stretching them, you get a better idea of how 3D strain affects cell morphology, physiology, and ultimately have a better in vitro model. And then also we have these synergistic effects. Basically, this is a program when you upload a video, it gives you a plot of your time versus contractile movement. Basically is a PIV analysis program that measures your displacement of pixels from your images and basically backs out your contractile movement, which also you can back out force generation, contractility frequency, your T50, which is time taken for your cell to relax, but basically all contractile measurements for myocardial cells. And so you can see here, this is just kind of an example of a contraction system where you have your baseline, then they introduce isoprenaline, which increases your contractility frequency, and then your carbachol here, which decreases contractility frequency, and you see you can basically measure that out using this program. So then ultimately future work, since we just started this summer, we're kind of building up all our mechanical suites that we're going to use as our testing platforms for these cells. So future work obviously would be combining all of this, getting our stem cells differentiated, embedding them in these collagen gels, placing, monolayers of your epithelial cells and seeing as a system how when you stretch all of this and you control all your mechanical factors, how they all work together to have a working in vitro model. So.
My name is Emily Despo, and I work with magnetic iron oxide nanoparticles for improved cancer drug delivery and cancer diagnosis. So one of the major problems with cancer chemotherapy drugs is that they attack all rapidly dividing cells in your body. So this means that when you take a chemotherapy drug, it attacks the lining of your stomach, causing nausea, your hair follicles, causing hair loss, as well as the cancer itself. So one of the ways that we hope to fix this problem, well, there's two major ways to fix this problem really, and one is to improve the selectivity of the drugs so that they affect only the cancer cells. And that would be something more like taking an antibiotic, which hopefully only kills the bacteria and not you, or to physically target these particles or the drugs to a tumor. So with our project, we're primarily looking at that first option, which is to make something that acts selectively on cancer cells. So one way to do that is to find something that's unique to the cancer cells, some sort of feature that they have. And so most cancer cells arise from normal healthy tissue, which means they have a lot of the same features, sometimes a little bit more, a little bit less, but it makes it very hard to find specific reasons or specific ways to target those cells. So there's a protein called survivin that's anti-apoptotic, and this protein is highly expressed in cancer cells across almost all kinds of cancer, and it's found at very, very low or negligible levels in healthy tissue, making it an ideal target to something that would only act on cancer cells, found across many types of cancer. And in vitro, antisense agents have been used to reduce this significant amount, reduce survivin expression by 30 to 70%, depending on the cell culture type. And with this reduction in viability, you also get increased sensitivity to chemotherapy, radiation, so different treatments, and even alone you can get reduced viability because the cell is more sensitive to the apoptotic signals from itself and the environment. The problem is that when you try to translate this in vitro success to an in vivo system, DNA is rapidly cleared. So any sort of DNA or antisense agent you introduce into your body will be cleared very rapidly, just like if you got a virus and you don't want it to go very far. So one way to improve this is to use a delivery platform. So in our case, we've picked superparamagnetic iron oxide nanoparticles, or SPIONs. And superparamagnetism is a very important feature. That means that they're not magnetic until they're in a magnetic field. So we can use them for something like an MRI, but up until we need them for that particular feature, they don't stick to each other in the bottle, in the needles, or in your blood vessels. So we start with this iron oxide nanoparticle, and it's coated in oleic acid, and then we coat it with a modified polyacrylic acid polymer. And this has hydrophobic oxylamine that will interact with this oleic acid, and it leaves carboxylic acid groups on the outside of the particle. We can functionalize these carboxylic acid groups using EDC to form an amine linkage with any sort of, or amide linkage with any sort of amine modified substance. So things like DNA, RNA, or antibodies. So in our particular case, we're interested in antisense to survivin, so the protein that I've just described. So an antisense agent, this is a 20-base single-stranded DNA that we attach. And when we do the synthesis, we do the whole thing, and so we start with the oleic acid-coated particles, which we then characterize to make sure that they're all the same size. And we found that this is a very uniform synthesis. They're approximately 7 nanometers. This is very reproducible. And using dynamic light scattering, we've shown that with the oleic acid coating, they're approximately 11 nanometers. We do the additional coating, and we don't have the size. This part of the coating is a little bit less consistent, so I don't have the exact size range. But we also want to know if these went into cells. So we used a fluorescent tag on the DNA label and incubated A549 lung adenocarcinoma cells with our fluorescently labeled particles. And at 24 hours, the particles were taken up by the cells. This is also with an endosome stain, so the endosome stain is red, the nucleus is blue, and the particles were stained with the green fluorescein. So all these areas of yellow, which you can see particularly well surrounding the nucleus of the cell, are areas where the particles have co-localized with the endosome. It means that they are being taken up by the cell, but it might also lead you to question whether or not they're able to have an effect. So we also have done some viability assays, but just to make sure that these particles that we were seeing were in fact inside the cells and not on the cells, even though we've washed them, we have confocal 3D renderings. So it's the same red for the cell, so cytoplasm, and green for the particles. And looking at the series of cutaways, you can see that the particles are in fact inside the cell, and there's holes here where the nucleus was, didn't get rendered, and you can see that they're not in the nucleus, which is good. They need to be in the cytoplasm to have their effect. So we did viability viability assays just to get an idea of how effective these particles were. And using a non-targeting control sequence of the same length of our antisense sequence as our control, you see that we have no change in metabolic viability after a 48-hour incubation with these particles. However, with our antisense agent, the viability decreases linearly with our concentration of the antisense agent. So this shows that the effects are coming from the antisense agent itself. And even though the effect isn't as significant on this graph as you might expect, it's because we have strong batch dependence on each time we add the DNA to the particles. So within a particular batch, these results are actually very, very focused and close together. So we're going to work a little bit on some of that consistency. One of the other ways we looked at their effects on cells was to look at LDH release. And LDH, lactate dehydrogenase, is an enzyme found inside of the cell, and it's released when the cell lyses. So this is a way of measuring cell membrane damage if it's leaking, if the cells have lyse. But LDH has a half-life of approximately 9 hours, which means we did this assay at 48 hours. If the cells died more than 9 hours prior to our experiment, there was probably not much LDH and not many cells in the experiment, or in our experimental wells. So this means that if you actually have no cells, if you've killed them all pretty early with a growth restriction of some sort, you can get either negative LDH release compared to a healthy control because there are less cells, or you have, your LDH release isn't necessarily where you would expect because of the cell number is different. So one of the things that you can see off of this, though, is that the non-targeting control doesn't change the viability hardly at all. While the LDH release, which would be increased cell death or cell lysis, increases with increasing concentrations of our spions with the antisense agent. One of the interesting things is that the way we did this was we used the same wells for both the LDH release and the viability studies. We took supernatant off to test the LDH, which means that we can come up with a direct correlation between the LDH release, with cell lysis in a well at that time point, and the viability at the same time point, which again gave us an interesting linear correlation, the number of cells viability increasing as DNA concentration increased. So at this point, even though these are sort of preliminary studies on this topic, we've shown that we can reproducibly make these particles. We get the 7 nanometer iron oxide core. We've also started characterizing the magnetic properties of this to see how it might be useful in an MRI. We've shown that our spions are taken up by cells, presumably by endosomes, but and they don't enter the nucleus, which means they're in the cytoplasm where they can have their effect. We don't actually see any changes in viability after 24-hour incubation, which is why it's not shown right here. But we do see significant changes after 48-hour incubation, and they're strongly dose-dependent. So we want to work a little bit on getting the consistency of our batches so that they always give us the same results. We're making one large batch so we can test them all for the same results. We also want to assess the changes in survivin expression throughout the cell's life cycle, or the cell's exposure to these antisense spions or the non-targeting control, and see how that correlates to the viability studies. And we've started doing this, and our initial studies have actually shown that the survivin expression is decreasing with our antisense agent. We didn't have enough data to show significance on the poster. And then we want to see if we can use these particles to sensitize our cells to chemotherapy agents like cisplatin. We have cisplatin have done initial dosing studies, and that's because survivin is anti-apoptotic, which means that its removal by itself does not inherently kill the cell. There still needs to be some sort of stimulus, whether intrinsic or extrinsic. So if we can stimulate this with cisplatin, we might see even greater cell death. And then we want to see how selective this is, because at this point our construct is really only therapeutically targeting these cells, and there's still a need for physically targeting these cells for improved MRI contrast enhancement, improved drug delivery capacity. And we have a couple of ideas for how we could possibly target this, actually using the survivin expression of the cell as sort of a marker because of the way that survivin in cancer cells is processed when it's overexpressed. And so this right here is just sort of the overview of this project. And right now we've focused primarily on this characterization aspect, we've worked a little bit on the composition, and we've started looking at the effects on cell function. And then hopefully, ultimately, we could start working on MRI contrast agent, looking at these, their drug loading abilities, their plasma protein interactions, and their biodistribution, probably in mice actually in the next couple of months.